This is a short video that will be going over the Bead cloning lesson, which is Lab 6 in Biotechnology I. I think this is a very useful exercise because I think it really solidifies some of the ideas surrounding cloning that are extremely important as we move forward for the rest of the semester. The last half of the semester is going to be all about cloning, and by working through this Bead cloning exercise, I think that you'll have a better comprehension of the different ideas that we will be actually working with pretty extensively as we continue through the remainder of the semester. So this Bead cloning exercise, while it's using silly little beads, it actually does a really great job of kind of making you understand what's happening throughout the cloning process. And so by working through this little exercise, it should really help you understand the difference between a restriction enzyme site or a recognition sequence of a restriction enzyme and actually digesting with a restriction enzyme. And I've kind of illustrated that a little bit here, where you can have a sequence, and in this case GAATTC is actually EcoR1 is the restriction recognition sequence. And so just having the sequence doesn't mean anything. It's not until you actually subject it to a restriction enzyme and that restriction enzyme recognizes its site and actually does its work and cleaves the two double-stranded pieces of DNA that you can actually say there has been digestion. So you can have a sequence and still have completely intact DNA, and then you can have a restriction enzyme actually chew that piece of DNA into very specific pieces. One of the other things this Bead cloning exercise is going to help you understand is some of the basic principles when we're talking about taking pieces of DNA and recombining them into a new molecule. This is a pretty heavy concept that I think the Bead really allows you to get your hands on and kind of get a more in-depth understanding of it. And so this is just a very general, you've got your plasmid DNA here, you've got a DNA fragment, which is very similar to what we're going to be working through, a DNA fragment of interest, and basically you're combining those into a single molecule that you can then investigate further. You will also kind of gain a better understanding of how restriction enzymes can aid in the generation of such recombinant molecules, how you can put in new pieces into plasmids, and additionally, because a lot of the cloning that occurs these days is actually a PCR-based technology, I want to make sure that we also understand that just the restriction enzymes and understanding the restriction enzyme sites is going to maybe help you verify clones or provide a screening mechanism in order to fully evaluate the work that you've done. And so the example that I have here is that maybe you have your original plasmid here, and perhaps in this original plasmid there's actually not an EcoR1 site. And then perhaps you've cloned in this nice little piece here into your plasmid, and it does contain an EcoR1 site. So while the remainder of the plasmid does not contain one of those restriction enzyme locations, once the insert is in there, it does. And so you could actually use EcoR1 then to screen for positive plasmids. Only the ones that actually digest with EcoR1 are going to actually contain the insert that you want. And so this can be extremely useful, not just in this case, but in a more general sense, that you can use restriction and restriction enzymes to really help you decipher what kind of cloning success you've had. And then additionally, I want you to understand, as everything is in Biotech 1, it seems like there's layers and layers upon layers of stuff. And so this is no different. And this activity, I really want you to be able to understand how it works. And by understanding how it works, you're going to be able to estimate, what fragment sizes should be based on, a plasmid map that you're going to be able to generate. And so just to kind of give you a little example here, if you have this pretend plasmid, and this plasmid, the total size of this plasmid is 2,500 base pairs. And usually when you're talking about a circular piece of DNA such as a plasmid, you kind of have this arbitrary, where there's an origin or something, you do have kind of a location that's kind of designated as base pair number 1. And some of the features of that plasmid are going to kind of be designated based on reference to that base pair location. And so you would have base pair 1 here, and then the one right before it is going to be base pair 2,499. So you're going to kind of go all the way around the circle, and at the end of the circle you're going to have that total plasmid size of, in this case, 2,500. So this can be extremely helpful. So if you have one restriction site here and you've got another restriction site here, these are usually designated with some of those location markers of base pair. So if this is base pair number 1 and your first restriction site is at 123 base pairs, and this is restriction site number 2 and this is at 945 base pairs, maybe these are the same restriction enzyme, maybe they're 2 different restriction enzymes and you set up a combination digestion. Whatever the case may be, you can absolutely determine what the fragment sizes would be if you cut with these restriction enzymes. And so by looking at the base pair locations here, you can basically just be taking this location here minus the 123 here, and it's going to tell you that the size of this fragment is actually 822 base pairs. And so this is kind of like some elaborate puzzle. And so from there, now you can say, all right, that's 822 base pairs. What does that mean? The remainder of this plasmid is going to be 1,000, my pen stopped working, 1,678 base pairs. So should you run this out on a gel, you would have 2 fragments. One that's going to be 822 base pairs and one that's going to be close to 1,700 base pairs. Easily distinguishable from each other, but if you have a restriction map, you can determine a lot of information, including how things should run out on a gel. Now, to actually get you into the pop bead portion of this, and again, I highly recommend that you read through some of the preliminary information that's provided to you at the beginning of this Lab 6. But I just wanted to kind of go over with you your beads. And so in your bead kit, you should have one linear set of beads and one plasmid set. And if they're not actually hooked together in the right manner, there should be a little sheet in there, in addition to the description in this lab here, where you can, I'd really strongly advise check everything before you start, because if your beads aren't in the right order, it ends up being a lot more confusing than it needs to be. So for your linear set of beads, you've got 13 white beads, followed by 2 pink beads, followed by another white bead, 2 green, 2 white, 2 red, 2 yellow, 4 red, 2 white, 2 yellow, 2 more white, 2 more green, and then 7 more white beads. When you lay this out, you want to have the whole end, which is the opening that could accept another bead, and then there's the knob end, which actually kind of has that little piece that fits into another bead. So double check and make sure that you've got your beads in the correct order, and if you don't, definitely reorder them. And then there also should be a circular set, which is a smaller set of beads, and these should be in the following order. Now, you can open them up if you want to verify, but you don't necessarily have to open up the plasmid if you don't want, but you just want to start with the special bead. There's a special bead that's kind of a twisted bead or an odd shaped. It's not like the rest of the beads. It looks different. And so counting from there is where you want to start. And then from there, you've got 5 orange beads, you've got 2 pink, another orange, 3 blue, 2 green, 3 more blue, another orange, yellow, and 2 more orange. All leading you back to the original twisted or strange bead. It's all different colors. Some of them have been white, some of them have been black. The set that I played with had kind of this purpley, shimmery bead, so there's just an unusual bead on there, and that's where you want to count from. So this is some of the information that's taken, pretty much directly out of the lab itself. And so you have basically what we're thinking about is that when you have 2 green beads together, we're considering that the green restriction site. When you have 2 yellows together, you're considering that the yellow restriction site. And when you have 2 pinks together, that is the pink restriction site. And so for the map then, for the plasmid, in this case it's going to be P-pop for pop bead, ha ha ha. This is our general plasmid map, and on that general plasmid we've got, a few restriction sites. We've got a green restriction site, we've got a yellow restriction site, we've got a pink restriction site, and then we have some other features that are kind of, if you look over the key here, kind of gives you some general information. And so there is one gene called the LAC-Z gene, and we're going to use blue-white selection when we do our cloning, but basically this LAC-Z gene being intact means it's going to produce a blue colony once it's in bacteria. Additional genes, there's another gene over here called AMP-R, and what does this mean? The AMP-R gene is actually a ampicillin resistance gene, and this allows whatever bacteria receive this plasmid, they would be able to grow on ampicillin plates. If they don't have this plasmid, the bacteria will die, and so it allows survival on ampicillin growth media. Then there's some additional notes here where you can, just like in that similar example I gave you a couple minutes ago, you can determine the different sizes of these fragments. And so from pink to green is going to be 750 base pairs. From green to yellow is going to be 850 base pairs. And then from yellow all the way back around to pink is going to be 1400 base pairs. And so by having a nicely defined plasmid map, it's going to allow you to know a lot about and be able to determine a lot about what it is you need to do next. Now, I know we're kind of giving you the map here, and it seems perhaps a little silly to have you draw it out again, but I really do advise you to go through the process of drawing out the map, because it's very easy then for you to double check it. But there is something about going through the process and actually drawing it out that is very valuable in kind of how it's solidifying in your mind. And so, draw it out, draw a circle, put your LAC-Z gene on there, put your green restriction site, your pink restriction site, your yellow restriction site, and maybe draw in your ampicillin resistance. It's important to go through the process, and you also want to be considering how this is looking based on, because we're going to be manipulating the beads, right? So you want to be able to understand how the beads are kind of translating into this hand-drawn map that I'm asking you to do. And so The special bead here is actually going to be the ampicillin gene. Any of these blue beads is actually part of this whole LAC-Z gene. When you've got these two green beads together, that's the green restriction site. And what I want to point out here is that even though the green restriction site is in the middle of this LAC-Z gene, or the middle of the blue beads, that is, the LAC-Z is going to be functional unless you've digested with the green restriction enzyme. So, draw a map, include some distances for what we've discussed already. Put the landmarks on there for like the ampicillin gene and the LAC-Z gene. And even though you've got, the result right here, please do go through the process and actually hand draw out the map. It will be amazing to you it'll make so much more sense to you in the end if you spend, just an extra minute here and actually go through and draw your own map. I believe this is going to end part 1 of this pop bead series, and so I'll catch you on part 2.